Hello, my name is Shanani and today I would like to talk about light microscope. What is light microscope? Light microscope is a biology instrument or tool that uses visible light to detect small objects such as microorganisms which cannot be seen with naked eye and enlarging them. They use lenses to focus the light on the specimen, magnifying it so that it could produce an image. The function of the light microscope is based on its ability to focus a beam of light through a specimen which is small and transparent to produce an image. The transparency of the specimen allows easy and quick penetration of light. The image is then passes through one or two lenses for magnification and viewing. And now let's look at the part of light microscope and its functions. First, eyepiece. It is also known as ocular lens. It transmits and magnifies the image from the objective lens. They form the final image which is projected into our eyes. Secondly, eyepiece tube. It holds the eyepiece and head or body tube. It connects the eyepiece to the objective lens. Nosepiece. It is a rotating turret that holds objective lens. The viewer will rotate the nosepiece to choose different objective lens. Stage. It is a platform where the slide is placed. Stage clips. The metal clips that hold slide in place. Arm. It connects the body tube to the base of the microscope. Iris diaphragm is used to adjust the amount of light that reaches the specimen. Condenser. It gathers and focuses the light from the illuminator on the specimen which is being viewed. Next is illuminator. Illuminator is a light source for the light microscope. Objective lens are the important part in the light microscope. This is because it is the very first lens that gathers the light from the specimen. It focuses and magnifies the light that coming from the slide. Several objective lenses that vary magnification and numerical aperture are mounted on the rotating nosepiece. The nosepiece must click into a place for the objective lens to be in a proper alignment. A standard microscope has 3, 4 or 5 objective lenses that in power range from 4 times to 100 times. Besides magnification, the objective lenses are used to improve the quality of the image. The coarse adjustment knob is located in the arm of the light microscope. It helps to bring the stage moves up and down to bring the specimen into focus. Fine adjustment knob is placed inside the coarse adjustment knob. It is used to bring the specimen into sharp focus under the low power lenses, and it is used for all focusing while using the high power lenses. We would like to proceed to know the method to use the light microscope. First, carry the microscope with 2 hands, one is under the base, another one gripping the arm, and place it gently on a flat surface. Secondly, plug the microscope into the power source and turn the revolving nosepiece so that the lowest objective lens is clicked into a place. This is also known as the shortest objective lens. The microscope slide should be prepared with the cover slip over the specimen. It could protect the objective lens if they touch the slide. Place the microscope slide on the stage and fasten it with the stage clips. Center it so that the objective lens is under the specimen. Look at the objective lens and stage from the side. Turn the coarse focus knob so that the objective lens will go downward without touching the slide. Now look at the eyepiece and adjust the illuminator or mirror and diaphragm for the greatest amount of light. Slowly turn the coarse knob so that the objective lens goes up and away from the slide. Continue until the image comes into a focus. Next, use the fine adjustment knob for a fine focusing image. Switch to next powerful objective lens to make final focus adjustments. And now you can examine your specimen. The proper way to use the light microscope is to look through the eyepiece with only one eye and keep another eye open so that it could avoid the eye strain. The things that you shouldn't do while using a light microscope is do not touch the glass part of the lenses with your fingers. Use the special lens paper to clean the glass parts of the lenses. When finished, raise the tube and lower the stage. Click the lower power lens into a position and remove the slide. Make sure the microscope is covered while it is not in use. Dust is the #1 enemy. And now I would like to explain the image orientation and magnification. The first lens which is closest to the specimen is being examined is the objective lens. Light from either external or internal source within the microscope body is first passed through the substage condenser, which will form a well-defined light cone to be concentrated on the specimen. The light passes through the specimen and into the objective lens, which projects a real inverted and magnified image of the specimen on a fixed plane that is within the light microscope, which is termed as intermediate image plane. The major function of the objective lens is the objective must gather the light from each of various parts or points of the specimen. Objective must have the capacity to reconstitute the light that coming from various points of the specimen into various corresponding points in the image. Sometimes it is also called as anti-point. The objective must be constructed so that it will focus close enough to the specimen and project real, inverted and magnified image up into the body tube. Essentially, a light microscope magnifies small objects and makes them visible. In the science of microscopy is based on the following concept and principles. Magnification is simply enlarging of the specimen. In compound lens, each lens sequentially enlarges and magnifies the specimen. The total magnification can be calculated by multiplying objective lens value by the ocular lens value. Thank you.